Thanks so much for having me, and thanks, Ed, for the great introduction to the brain. These are indeed exciting times for improving our understanding of the brain. I'm going to talk about our efforts to understand brain architecture and brain development. And since this is beyond the cell atlas, I'll try to put my take on what I think is the next step for a cell atlas. This is sort of the classical, by now, approach to atlasing. We use single cell methods to take a sample of cells out of some brain region. We do single cell RNA sequencing, and then we cluster the cells. And this has turned out to be an extremely powerful approach to understand the composition of complex tissues, in particular the brain. And so I'll begin with our latest effort in this direction, where we try to survey the whole mouse nervous system and to get sort of a global big picture view of the nervous system. This is kind of the opposite approach to what Ed just described. It's broad, but it's not super deep. And let's see what we found. This was work led by Amit and Hannah in my lab and a number of collaborators. And I want to give a shout out also to a paper that came out at the same time from Steve McCarroll's group with Eva Makosko and Arpai Saunders that used similar methods to study the brain. So I will just do a quick overview of this work because I want to get into talking about dynamics. So in this work, what we did was we tried to sample the whole brain. So we dissected the brain into regions that were non-overlapping, but also not leaving anything out. So at least in principle, we could discover everything. Of course, that's a sampling problem, but we didn't intentionally leave anything out, with the exception of the retina and the inner ear. And we also included the major parts of the peripheral nervous system. So we have the sensory and the sympathetic and the enteric nervous system as representatives of the periphery, and missing therefore the parasympathetic. And this is based on about 500,000 cells using droplet sequencing. So, one thing that I think we can learn from such analysis is sort of the global organization of the nervous system. So this is a dendrogram showing all the 265 cell types that we defined. These are pretty conservative cell types. These are sort of broad, well-defined cell types in most cases. And this dendrogram is made just by looking at transcriptional similarity. So it's a dendrogram that shows similarity in gene expression. Right, and what you see is that first of all there is a perfect split between neurons and not neurons. So the major difference that comes out in this data is whether you're a neuron or not. And this is because neurons express a very large number of genes that are neuron-specific, involved in cytoskeletal proteins or synaptic proteins and all of these things, that the ion channels and so on that are required to be a neuron. And that's actually not really a developmental split. There is no subtree of the embryo that makes all the neurons. That's not how it works. So it's more a matter of a phenotype. Maybe it's an evolutionary thing, but it's definitely not developmental. But if you go to the next step, you see that the next level splits central nervous system from peripheral nervous system. And this is clearly a developmental split because the CNS derives from the neural tube, whereas the PNS derives from the neural crest that migrates out and populates the body. And we see the same kinds of patterns if we go into the depth of this tree in the CNS, for example. The major subdivisions here are anterior-posterior regions. So you have the telencephalon here, so part of the forebrain. You have the diencephalon and the mesencephalon here. Those are the sort of the midbrain. And you have the hindbrain here, and you have the spinal cord. So these are segments of the nervous system from front to back. And these are also developmental divisions because the nervous system develops by initially patterning along the anterior-posterior axis. And then inside of these trees, you see a split between excitatory and inhibitory neurons most often. This is true of the cortex, as Ed pointed out. So all of these are the excitatory cortical neurons, whereas the inhibitory neurons are here. Right? So they segregate, and it's again because they have different developmental origin. And the same is true of the spinal cord, for example. There's a perfect split between excitatory and inhibitory. So I could talk about this forever, but I'm going to charge ahead. I added 2 slides about astrocytes, since we are on the topic. It's indeed true, as Ed said, that classically we distinguish 2 types of astrocytes in mice, and also I guess in human. And then there are some regions that have their own special astrocytes, like retina has Müller glia and cerebellum has Bergman glia. But overall, there is this distinction between fibrous and protoplasmic, or we can use those terms. So we were surprised to find actually a large number of regionally specific astrocytes. So this is a magnification of a part of the tree that you saw in the previous slide, and we found these, I guess it's 7 types of astrocytes, including the Bergman that were completely known before, but these other types are we believe, novel. And so if you look inside of the cortex, you have what we call telencephalon astrocytes that are spread out across the telencephalon. So those are in this Disney plot here, shown in this cloud here. And there's a split between 2 types here that correspond exactly to the protoplasmic and the fibrous, or the GFAP high and the GFAP low that you saw in the previous talk. But these are telencephalon specific. So if you go to another brain region, you will find other types of astrocytes that are distinguished by distinct markers. So for example, in the olfactory bulb, we found olfactory astrocytes that are clearly distinct from those of the cortex. It turns out also that the brain from the diencephalon and further posterior, so diencephalon, midbrain, hindbrain, spinal cord, they have their own astrocyte that's shown here. So this is the other big cloud. So there's a clear distinction between the telencephalon and everything else. And we think again that these are developmentally specified astrocyte subtypes. And this can be shown in various ways, but one way is to look at transcription factors that are responsible for and involved in anterior-posterior patterning. So FOXG1, for example, is a transcription factor that's activated specifically in the forebrain, and indeed it labels all of the telencephalic astrocytes, but not the non-telencephalic astrocytes. And vice versa, if you take transcription factors that are specific for midbrain or hindbrain, those are found in the other type of astrocyte. We can confirm this by staining. So up here is a summary of all the different stainings we did to confirm this, and you can see by different combinations of markers how distinct types of astrocytes are located in distinct parts of the brain. And I think it's amazing that there is this incredibly sharp border between telencephalic astrocytes and non-telencephalic astrocytes, which really, again, I think reinforces the point that these are developmentally specified. And this is exactly the border between the telencephalon and the diencephalon that's established very early on in the embryo. And apparently these astrocytes don't like to move around enough to mix there. Okay, but the future of atlasing, I think, is going from static pictures to dynamic. And what I mean by that is well illustrated by this drawing from Del Rio Ortega. He was the guy who discovered and named oligodendrocytes. This drawing, I think, is a beautiful summary of brain development that shows the major lineages of the brain going from the neuroepithelium. So this is like a layer of cells early on that forms the neural tube, from which cells emerge first making neurons down here, and then making astrocytes down this path, and eventually oligodendrocytes that wrap around the neurons. And he has drawn this summary of the dynamics of brain development based on his observation of static pictures. He had only the microscope as his tool. And we're kind of stuck with the same, although we have extremely powerful molecular tools, they're still static snapshots that do not give us dynamic pictures. So I want to talk about our efforts to extract dynamic information from static snapshot data, particularly RNA-seq data. This is a collaboration with Peter Khaychenko and led particularly by Joel Elamano in my lab. So this comes from the observation that developmental processes and dynamics occur on a time scale of hours to days. So cell cycle is maybe 12 hours in early development, and differentiation processes also take place during hours and maybe a few days. And this is not so far away from the timescale of RNA metabolism. So by which I mean transcription, splicing, and degradation. So half-lives, for example, are on the order of 10 hours. And the rate of transcription and splicing are such that the flow through this life cycle of mRNA is on the order of an hour or a few hours. But this means that the RNAs that are mature, that we can observe when we do RNA sequencing, reflect, let's say, the current state of the cell, whereas the unspliced, immature, nascent RNAs reflect what the cell is going to be in a couple of hours. Right? There's a time differential there. So if we could exploit that, we could actually learn something about the future of the cell. So this is how it would work. Imagine you have a gene that's off, and then it's switched on, and then off again. You would have a rise first of unspliced nascent RNAs to some steady state. And then spliced RNAs would also rise, but there would be a time delay. So there is a little bit of a time lag here, and vice versa when the gene is switched off. And if we plot this as a phase portrait, spliced versus unspliced, what we will see is that we can distinguish the part where the gene is induced from the one where it's repressed by the ratio of unspliced to spliced RNA, basically. Right? So we can see whether the gene is going up or down, telling us something about where the cell is going in gene expression space. So first question is, can we detect unspliced mRNA in single-cell RNA sequencing data? And the answer is yes, for all common methods. Here are fractions of unspliced reads in SmartSeq2, InDrop, and 10x Chromium, and it hovers around 15–20%, something like that. And so this is actually not a trivial amount, and it's something to work with. So actually that's very surprising, because all of these methods are based on oligo-T priming to poly-A tails. It turns out the reason that this works is that there is very abundant sort of internal mispriming in all of these protocols from internal poly-A or poly-T stretches inside of introns. So here's a zoom in of a genome browser of a gene. Here's an intron, here's an exon, here's the next intron. These notches here show where there are poly-A or poly-T sites. And you can see that 10x Chromium, for example, gives a nice peak upstream of every poly-A site. So these are not poly-A tails, these are poly-A sites that are encoded in the genome. I think these are at least 12 bases long, that's the criteria. But it's true also of InDrop, and it's actually true also of SmartSeq2, although it's more smeared out, because in SmartSeq you prepare not just the end of the transcript for sequencing, but the whole thing. And you can summarize this, and you get these nice peaks upstream or downstream relative to the orientation of the poly-A stretch. All right, so that explains why it works. The next question is whether these products, being kind of artifacts of the protocol, really represent nascent RNAs. And to investigate this, we did a 4th-IUU labeling. So this is metabolic labeling. We took cells and incubated them for 5, 15, or 30 minutes with 4th-IUU. It gets incorporated in nascent RNA. And then we performed, so normally you would do this, and then you would do an RNA sequencing using a protocol that extracts all RNA, not just polyadenylated. But here we did specifically, we used a single-cell RNA-seq protocol that uses oligo-T priming to see if we could actually detect the nascent RNAs. And the expectation would be that this acts as a sort of virtual experiment where a gene is switched on at time point 0 and it stays on, and you would approach a steady state in unspliced and spliced and so on. And indeed, for most genes, this is exactly what we see. So we can see the ratio of unspliced to total RNA going down as the unspliced RNA gets converted into spliced RNA, and vice versa. This also allows us to fit fit the model of transcription that we posited. So this simple model to this data, and to estimate the parameters. And in particular, we can estimate the time constant of relaxation. So how quickly do genes reach their steady state levels? And it's, of course, very variable, but roughly on the order of an hour. So this again indicates that we have a time constant of about an hour. We might be able to see an hour into the future. So applying this to, now again, bulk RNA, this is circadian rhythm, this is liver cells. this is a data set where RNA was sampled every 3 hours, and you can see spliced and unspliced RNAs. We separated them. And the important point here is that at every time point, the unspliced RNA is more similar to the spliced RNA of the next time point. Right? So if you compare, for example, here, the unspliced RNA at time point 12 looks more like the spliced RNA of time point 15, again pointing to the fact that nascent RNA points to the future of the cell. And in this experiment, it looks like we can see about 2 hours into the future. Of course, again, with a lot of variability. Looking at individual genes, we can plot these kind of phase portraits, spliced versus unspliced, and indeed they show this behavior that we predicted from the model. As they are being induced, they go above the diagonal, so the unspliced is leading, and as they are being repressed, the unspliced is, well, again, leading, so therefore you go below the diagonal. You can also combine all genes, so you can do a PCA plot and actually do an extrapolation from the current state to the future. And so this is a diagram that shows these different samples in PCA space with an arrow pointing to the predicted future state based on all genes. And now you can see that each time point points again towards the future, so you get this sort of clockwise portrait. So this is what we call RNA velocity. RNA velocity is the time derivative of gene expression, and it's an arrow that points to the future state of the cell in high-dimensional expression space. So now we're applying this to single cells. So here is an experiment with cells that differentiate from a sort of stem cell state to a mature state called Chromaffin cells. And you can look at genes that are high in the beginning or high in the end, and you can see that they indeed behave in this phase portrait plot exactly like we predicted, although now the data is much, much noisier because now we're at single cell level. And this allows you to estimate velocity, so you can say that, for example, here the velocity is high and then it's low, so it's being induced and then repressed, exactly like our model. And now this allows you to put an arrow on every individual cell, so you can see the collective motion of cells across this PCA space. It's important to understand that RNA velocity is a high-dimensional vector that gives you the behavior of all of the genes of the cell. And so there might be independent processes that have velocities that tell you different things about the cell. For example, here in PC components 1 and 2, you see differentiation from stem cells to mature cells. Whereas in PC4 versus PC5, we think that you're seeing cell cycle. So the cells are cycling, and therefore there is velocity also around the cell cycle. And this is important to keep in mind when interpreting this, that you're seeing sort of superimposed multiple processes. What I showed you now was SmartSeq2, but it also works with droplets. This is in-drops. This is a reanalysis of a published experiment by Michael Greenberg's group, where they did light activation of cortical cells. So we have 0, 1, and 4 hours light stimulus, and then they took out the cells and did in-drops on this. So we reanalyzed this data set, and you might expect something like successive waves of gene activation due to this light stimulus. And indeed, what you see is that the cells sort of range in a triangle. You have the unstimulated cells here, you have the 1 hour and the 4 hour here. And superimposing RNA velocity, you can see that at the 0 hour there is no velocity. They're not going anywhere because they have not received the stimulus yet. At the 1 hour, they've already moved here in gene expression space, and you can see that they're going towards the 4 hour point. Whereas the 4 hour point is here, they are actually going back towards the 0 hour. So again, pointing to these successive waves of gene activation in response to the light stimulus. So I guess I'm running out of time, but let me justify one of the key assumptions here. One of the key assumptions is that the degradation rate is constant for each gene. So we fit a constant degradation rate, and we assume that only the transcription rate varies. So to investigate whether this is reasonable, we actually measured or estimated degradation rates in a large cohort of tissues. This is single cell data from the Tabula Muris consortium that Spiros Darmanis presented yesterday. And fitting now spliced versus unspliced in these, what we believe are terminally differentiated cell types. So these are more or less stable cell types that are not going to differentiate further. At these steady states, what you would expect is that if degradation rates are constant everywhere, then the ratio between spliced and unspliced would also be constant. And indeed, for most genes, we see that all of these tissues fall on the same line, or the cell type fall on the same line, pointing to a fixed degradation rate. There are some cases where there are actually very low correlations, and those cases are often very well fit by two degradation rates. So for example, here you have a high gamma and a low gamma. And so we think in those cases there is something that differentiates the degradation rates in different tissues. And one thing that can happen is that there are splice variants, so there are different versions of the gene active in different tissues, leading to differences in these constants. So let me show you then quickly how to apply this to a branching lineage. This is the hippocampus development, which starts with the radial glia and branches out into different kinds of neurons, or astrocytes or OPCs. And applying velocity to this, you can actually start to see more or less how the cells differentiate during development. So there's a starting point here, and you can see all the arrows on this side point down towards astrocytes or towards OPCs, and then they come to a rest here. Whereas if they decide to go right here, they go up here, and then they make a choice and either become granular cells or different types of pyramidal cells. So you can sort of follow differentiation as it happens by following the arrows here in this manifold. And here is a choice between different types of pyramidal cells in the hippocampus. And summarizing this, you can really see a velocity field of how the cells move along this manifold. Let's just finish with this one. So one of the, I think, what's most exciting about this for us is that it's a type of analysis that is more or less a measurement of the dynamics of gene expression. It's not an inference really, although there are many assumptions, but it's really measuring something in the data that gives us information about dynamics. And it's based solely on RNA sequencing, single-cell RNA sequencing, which means that it also works in humans. So this is a plot that shows cortical maturation from the progenitor, the radial glia, differentiating into more or less mature neurons. And you can see that we have a nice velocity field across this that tells us the direction here. And of course, this is not a branching pathway, but as we start to collect larger and larger data sets from developing human brain, we expect to be able to reconstruct basically the differentiation lineage tree of the human brain using this kind of data. And by that, I will finish and thank all the people in my group, our great collaborators, especially Peter, our funders, and we try to share everything, put all the papers in my archive and all the data online and all the code and everything. So check out our website. The mouse atlas is at mousebrain.org, and the velocity tools and data and notebooks and everything is at velocito.org. That's cyto as in a cell, right? Thank you.
Great. We have time for maybe 2 or 3 short questions.
Thanks. Okay, so Sten, we talked about this, and you know that I'm a huge fan of the velocity. But I still want to make a comment regarding your gamma or your turnover rates, which you say you find largely constant. So, you know, 3' UTRs are incredibly important for cells. In humans, they're huge, and they're bound by hundreds of RNA-binding proteins and microRNAs, which a lot of them regulate turnover and stability of the messenger RNAs. So I think this is also probably a little bit important. And now I think yes, you have some examples there, but I think the point is more that if you choose a system which is clearly a system like early development, where transcription is just driving lots of or probably almost all of the decisions, then you can estimate your future of cells very nicely with the kind of assumptions. But I think if it comes to polarized cells or cells which are post-mitotic, you may have a difficulty in applying the same assumptions. This having said, again, I'm a huge fan of this, and I think actually we will be able to measure all these constants, so I think these arguments will be historic at some point.
So thank you very much for the kind words. I want to make 3 quick points. So first of all, I agree. We definitely present, essentially we present a method that has 3 unknowns and we're measuring 2 things. So we have to make some strong assumptions to make that work. And we fully agree. And I think that there are ways forward by which we could actually measure these things rather than make the strong assumptions. Second, I want to say that I showed you these examples of genes that have different gammas in different tissues explained by splice variants, but these are actually the minority. We had to hunt to find these cases. Most cases where there are 2 gammas, we don't understand, and I'm sure that one part of the explanation is that there are tissue-specific or organ-specific RNA binding proteins that affect degradation rates, and there's at least 15–20% of the genes that show this phenomenon, so it's clearly significant. Having said that, this is post-mitotic cells that we're looking at here, and I think that during development, I agree with you that it's more even more likely that transcription is the driving force rather than degradation, just because of the kinetics. It's hard to control things fast with degradation.
Yeah, hi. So congratulations for the excellent work. And you said in the beginning that this approach works because there are like naturally occurring poly-A sequencing within the introns. So I was wondering whether you checked if the frequency of this occurrence is the same genome-wide or not, and if not, whether it is introducing some kind of biases, meaning that some genes which do not have this poly-A sequences, then they do not contribute to the velocity computation.
Yeah. This is a very good point. So it's important to realize that this works collectively when you look at the whole transcriptome. But for individual genes, it's hit and miss, and it's more miss than hit, because for this to work, you need to have enough intronic reads. So the introns have to be long enough. They have to contain poly-A tails or poly-A stretches. And just the kinetics have to be such that you can distinguish the unspliced from the spliced. So in a typical experiment, on the order of 2,000-ish genes are usable out of, let's say, 8,000 or 10,000 that are expressed anywhere in your whole experiment. But the differentiation processes or the activity-dependent transcription that we're looking at usually involves many genes, many tens of genes at least. So it's actually enough to measure 1 or 2 of those in order to be representative of that process and visualize it. But again, if you go and look at your favorite gene, it's actually not likely that it's going to work, or it's more likely that it's not going to work than it's going to work.
One really quick last question.
Yeah, I enjoyed Sten.
Hi, Sam.
Many recent experiments show that there's oscillations in the nucleus, and the methylation level oscillates. Long-Kai will talk about the oscillation in splicing. I'll talk about the oscillation of transcription factors. Always cycle time shorter than the cell cycle, a few cycles in a cell cycle. So I wonder, do you think what you see is related to that or
so
not?
I would say one of the nice things about RNA velocity is that it doesn't make assumptions about the structure of the high-dimensional manifold. So things can go in cycles, for example. That's perfectly possible. And you would see, like I showed for the cell cycle. And a process that, like the cell cycle, we can definitely detect it, and we can see the cells going around in a cycle. But if it's a process that's much faster than the cell cycle, if it's a 10-minute oscillation, that would be very hard. Yeah, so there is some threshold by where the frequency is just going to be too high relative to the relaxation time of splicing and degradation that's involved in these measurements. And it will be smeared out, and we will not be able to detect it.